Good morning, everyone. So I'm Cynthia Bryant. I'm the Chief Business Officer at Demetrix, and really happy to be here today to talk a little bit about what we do and the research that we're performing. It's a little bit different than I think a lot of the other companies here. We're not a plant-based grower. So let me tell you first about Demetrix, and then we'll get into the rest of it. So at Demetrix, the company was founded by a gentleman, Jay Kiesling. He's a UC Berkeley professor who started several companies in biotechnology that are dedicated to producing rare ingredients to help promote human health and wellness. And at Demetrix, we provide affordable, sustainable access to rare plant ingredients for healthier lives. And we're specifically focused on rare cannabinoids. So the way that Demetrix operates is on these pillars of ensuring that we have high purity product, so 98% pure or better. We produce in a very sustainable fashion. Biotechnology uses a lot less resources than typical agriculture when you're talking about rare ingredients. And everything we do is science-backed in terms of the research that we perform as well as the technology we develop. And these three things are what really drives our innovation. And by innovation, what I'm really speaking to is the desire to find new ingredients to help people and to then design production processes that can bring those ingredients at high enough volumes and high enough purity to be available and affordable to those that are in need. So how do we do this? So if we just take the cannabis plant, there's over 100 different cannabinoids in this plant. But the fact of the matter is most of these cannabinoids are in such small quantities in the plant that it is just impossible to be able to isolate them at the volumes needed to properly research them. And this is where biotechnology comes in, because what we can do is we can produce single cannabinoids completely agnostic to how rare or common they are in the plant. And we can produce them at high volumes and high purity so that they can become available for researchers to do research on these specific isolates and understand what the individual cannabinoids contribute in terms of beneficial properties for many indications. Moving on, this is a picture of our labs in Berkeley, California, where we're headquartered. And so we use fermentation to produce these cannabinoids. So we take a microorganism such as yeast, baker's yeast, commonly used for beer and producing bread, and we program it and say, we want you now to make this specific cannabinoid. We develop a very controlled production environment in a bioreactor, which is a fancy word of saying a fermentation tank. And we grow these yeast, and as these yeast grow and as they are in an environment that's conducive, they start to produce that cannabinoid, and then we purify it from the fermentation broth and we have our product. So the benefit of this, especially when we're talking about rare ingredients, is the reduction in water, energy, and land use. Because no matter if we're talking about a cannabinoid or a fragrance ingredient, that particular plant has to produce an entire plant. It has to produce the roots, the leaves, the stems, sometimes the flowers, etc., as well as that specific ingredient we're after. And if that ingredient in the plant is very rare, you're creating a lot of added biomass, a lot of energy input, a lot of water utilization for just that small component. And we take all of that out because all we're doing is using a small yeast, and actually over 1,000 yeasts can fit into a trichome on a cannabis plant, and we're telling it only to make that specific rare cannabinoid. So I know everybody here is very aware of the endocannabinoid system. When you hear about cannabinoids, a lot of times what people talk about are the CB1 and CB2 receptors. What we've been doing with our technology is we now have over 40 different rare cannabinoids that we have been taking through various research to understand and catalog what they actually do from a human physiology perspective in terms of the receptors that they interact with, and also taking them into in vitro and a little bit in vivo tests to then prove to understand, do they actually manifest into the specific benefits that we hope that they do based on the indications that we see through the receptor screening. And so what I'm going to do for the rest of this presentation is talk a little bit about one specific cannabinoid out of the 40 that we currently have under research, and that's CBG, or cannabigerol. And so we recently commercialized cannabigerol as Arete G, and with our partner Evonik Industries, which is a large German ingredient supplier, are now bringing this ingredient to the market for personal care use. So there's a couple of things, there's a lot of research out there about cannabigerol in general, and there's indications that it's a very strong anti-inflammatory. We also see that it has antimicrobial effects and strong antioxidant properties. But a lot of this, then we have to do further research to understand, does that translate into an actual claim? Because a lot of this is done at receptor or very preliminary screening levels. So the work that Demetrix did was specific to our cannabigerol, Arete G, which is 98% pure. It has 0 THC in it. Our process is completely incapable of producing THC. Has 0 terpenes. So this is a very clean isolate that we've taken into this research. And what we've seen when we took it through is initially it was GPCR and ion channel screening. And we saw strong anti-inflammatory properties. We also saw that it was a strong antioxidant. And we saw that it had interesting antimicrobial potential as well. But the next step that we did is we put it into in vitro skin assays to understand, does that actually translate when you start to go into industry standard testing? And along with it, we took a couple of other cannabinoids that we have listed on here through specific in vitro tests. And what we saw is that, first of all these cannabinoids are physiologically active. And I think that that's really important because there's a lot of anecdotal evidence out there about cannabinoids. But having actually screened through the entire library of GPCRs in the human body, 40 different cannabinoids, I can sit up here and assure you that they do interact with the human body. We also know that they interact differently. Each one has its own unique fingerprint. They do not all do the same thing. And I think that that's also another important fact and discovery that we've made, because they're not the same. And what that leads us to believe is that there's a tremendous amount of potential, not only as individual cannabinoids, but to expand research and understand the right combinations of the right cannabinoids to be able to help people with specific indications. And we're just scratching the surface here in terms of the amount of research that still needs to be done to really harness the true potential of these ingredients. So here, this is tip of the iceberg in terms of what we've done and also where we need to go. So in these in vitro assays, what we see is the darker color is stronger, indicates stronger performance, lighter, less performance. And you can see here, even among these handful of cannabinoids, we see varying differences. The other thing that I'd like to point out is that we saw that CBG was a really strong and interesting antioxidant in basic ORAC, DPPH tests. But when we put it into antioxidant in vitro skin assays, we didn't see that it was a very strong performer. And I think that this is also another important thing in terms of taking the research to the next levels to really prove out and to elucidate how it translates into that actual benefit that people need. So with that, also went a little bit deeper in terms of looking at these specific assays and understanding the anti-inflammatory effect of CBG on skin. So what we did here is, I just want to get it all up. There we go. So we ran this in vitro assay where we took normal human dermal fibroblasts and normal human epidermal keratinocytes. And the first thing we did is we ran cytotoxicity assays to understand what's the highest dose that we could actually use of cannabigerol as well as some other cannabinoids in these tests. And the first thing that was interesting is that they are different in their cytotoxicity. So we ran these tests several times just to make sure. CBG is less cytotoxic than cannabidiol, CBD, by quite a significant amount. And you'll see that translated into some of the charts that I'll show next. Once we understood the cytotoxicity, we then were able to determine what the right concentrations would be so that we wouldn't destroy the cells in these assays. And through a plate model, put the Areta-G in with a stimulated inflammation of these cells, and then understood through a positive and negative control the performance of the CBG. So the first one we did was a PMA stimulated model. And what you see in here is the percent inhibition. So you want to inhibit PMA in order to, you want to inhibit IL-8 in order to see an anti-inflammatory reaction. So IL-8 is one of the key cytokines in terms of inciting inflammation. The blue bar that you see is actually the positive control, and that's staurosporine. So this is a known anti-inflammatory that works very well. The red bars that you see next to it are the percent reduction of CBG. So you can see at the higher concentration, which is 3 micromolar, CBG was right at the performance basically of staurosporine. And we had a really nice dose response curve with this one. Now the gray bars next to it are actually cannabidiol. And what you'll see there, if you look on the x-axis, is that the concentrations are different for CBD versus CBG, and that's just reflective of the cytotoxicity differences between the two molecules. But what we found was very interesting is that we hear a lot about CBD being an interesting anti-inflammatory, and a lot of people translate that into skin health. But what we saw in this industry standard skin model is that CBD really wasn't a good anti-inflammatory in terms of other cannabinoids. Now, what I don't have on here is the other ones, but CBG, I'll just share with you, was the most highest performing out of the lot. So the next test we did was PGE2 in PMA stimulated cells. And here, the positive control that you see is indomethacin. And what we see here is also a decent, I would call it, a dose response. What we think is we just hit the upper limit of the CBG response, which are the red bars next to it. But again, strong anti-inflammatory properties were seen in this model. CBD in this model showed absolutely no response in terms of preventing inflammation. So the other thing that we wanted to make sure is, are these products safe? And so we did a lot of skin irritancy testing. These were in vivo. And what we found is that CBG and CBD were both non-sensitizers to skin. They're also not ocular irritants or in any way damaging to the eyes. So that's all that this chart is really showing. And it was interesting because we had over 100 test subjects and not a single one of them showed any indication of irritation with CBG, which is pretty rare. So we were very happy with those results. Now, the other thing we did was minimum inhibitory concentration assays against different microorganisms that are interesting ones for skin health. So the first one that we did here is for skincare is C. acnes. And there's a lot of research already on the efficacy of various cannabinoids on C. acnes. What we saw was very similar to that research, so we feel that we've got a good validation on that. The next one are actually 2 oral care targets, which we were surprised to see. So S mutans is a microorganism that is strongly linked to periodontal disease. And it's also a very difficult microorganism to control with natural products per se. So the oral care industry uses chemical ingredients to help to control S mutants in toothpaste, mouthwash, et cetera. And the fact that we now have a potential natural ingredient that not only showed efficacy against this, but also was not broad spectrum. So these cannabinoids are not broad spectrum in terms of their antimicrobial activity. They're actually quite measured and quite specific. And that's really important because we're all very aware of antibiotic resistance and also wanting to cultivate a healthy microbiome, not only on our skin, but also in our oral cavities. The other thing that it was good is that it was active against P. gingivalis, which I'm sure all of us are familiar with in terms of oral care. So here we have an ingredient that's naturally derived and hits 2 of the key oral care microorganisms that we want to control for proper oral care health. Now, when we layer on top of this the fact that CBG was also showed a strong anti-inflammatory response against IL-8, it gets even more interesting for oral care because there's research showing that IL-8 inflammation is a link to chronic periodontal disease. So not only do you have to control the microbiome, but you also need to control the inflammation. So really, really interesting results with this. The other one is personal care, and here M. furfur was an interesting target that CBG showed efficacy against. M. furfur is one of the funguses that is often linked to dandruff. There's 3 of them, M. furfur is one, and depending on what region of the world you live, M. furfur is either more important or less important in terms of controlling the dandruff. So mainly Southeast Asia is where that's really important. But it is also one of the key reasons that some people suffer from tinea versicolor. So this is the skin condition that often shows up on the backs where people have white patches on their skin. And this can be a very difficult condition to treat, and to have yet again a natural ingredient that shows efficacy against this M. furfur fungus shows a lot of promise to help people not only to look good, but also to feel better about themselves. So in summary, what we're seeing specifically with cannabigerol or Rete-G is that it shows a lot of promise for aiding and being a good bioactive for skin care, not only for what I'll call the more cosmetic concerns in terms of anti-aging support. A lot of anti-aging or a lot of the aging from our skin is an inflammation response. So if we can control the inflammation, we can slow down the aging process, is the theory. But it can also help in some other conditions that, quite frankly, probably have a lot stronger or greater impact on how people feel about themselves and their well-being in terms of helping with acne, also, as mentioned, tinea versicolor, and then oral health. I can't stress enough how important oral health is. It's linked to not only, diseases within the mouth, but it's also now being linked to certain CNS concerns with things, microorganisms potentially going through the blood-brain barrier and through the oral cavity and creating issues with mental health. It can also be linked to digestive support as well. So being able to balance the microbiome in our oral cavity has wider range benefits than just having a pretty smile. As mentioned, we are looking at other cannabinoids. We have over 40 that we're taking through many of these various assays, and also we'll be taking into clinicals with our partners. And what I'd like to just leave you with is that I'm super excited about where we are today in terms of helping people with health and wellness as an industry. This plant has so many interesting ingredients in it that hold so much potential to really help people in a safe and an effective way. And I think that as an industry, we owe it to the consumers around the world to do the proper research and to provide products that we know will work time and time again and understand how they work and why they work, so that we can provide real solutions time and time again for people that are in need of that. So we're always happy to share more about the research we're doing. We believe that education is key in this industry and that we need to stand on each other's shoulders, so to say. So we welcome conversation and for people to reach out to us to learn more about what we're doing and our findings. We will also be communicating a lot of these as we learn more, and look forward to speaking with you all about what we're doing in further detail. Thank you.
Hi, very interesting presentation. Of the 40 some odd cannabinoids that you've isolated, are you able to say how many are identical to those produced by cannabis sativa as opposed to new chemical entities?
They're all 100% identical. So every single cannabinoid we produce can be found in the plant. They're just in such small quantities, we can't physically isolate them from the plant, but they're all natural.
Okay, thank you.
Wonderful talk. I was just wondering if you were able to look at the acid forms of some of these, or are you just producing the neutral forms?
I'm sorry, I had a hard time hearing you.
Sorry, I was wondering if you were able to look at the acid forms of some of these compounds, or you're only able to produce the neutral forms?
No, we can produce both. So we are looking at both acid and neutral forms, and I will say the acids do behave differently than the neutrals, physiologically speaking.
Does neutral mean decarboxylated?
Yes.
Okay, I'm just checking. The CBG for the clinicals that you did, do you know what percent concentration, or do you have a recommendation for a percentage of concentration for a product application?
Yeah, so in skincare specifically, you usually don't want to go above 1%, and this is consumer product skincare. So we're currently doing clinicals to see its efficacy at that 1%. But for an OTC or pharmaceutical, depending on the formulation, you could go higher, but the clinicals would need to be done to check the actual efficacy within the formulation.
Thank you.
Yeah.
Hi. Are the commercial products, are they topicals, or are they something that you would ingest orally?
Yeah, so right now, our partner Evonik is specific to topicals, personal care, but we are currently working with the FDA as well as EFSA to get the proper approvals for ingestible products.
Hi, thank you for the nice talk. I was that you had very little reactions, like negative reactions when it was on people's skin. Did you have any issues with it clogging pores, or the molecules the right size so that it's not going to cause any, it might reduce some breakouts when I produce some, or how is that?
Yeah, we did not see any issue with clogging pores. These are fairly small molecules. It really depends also, though, on the formulation that you put the cannabigerol in, because cannabigerol itself is a solid, so you have to solubilize it in something to apply it to skin, and depending on what you solubilize it in, then maybe you can get into comedogenic issues, but the cannabigerol itself is non-comedogenic.
Awesome, thank you.
Thank you so much for your talk. What was the method that you used to measure IL-8 and PGE2 levels?
I believe it was fluorescence. I'm looking at ELISA. I'm sorry, it was an ELISA.
Thank you so much for sharing your work. That was really enlightening. I had a question about the 2% that's left after the 98% CBG. I wonder if you've fractionated and taken out the CBG to look at efficacy, as we saw last night with Duddy Mary's talk. Sometimes that really low percentage can be really effective as well.
Yeah, so the key thing to keep in mind is that we don't use any plant material. So the 2% that's left in our product actually comes from the yeast that's used to produce the cannabinoids. So it's mainly things like carbohydrates and proteins. There's no terpenes in it or other cannabinoids except for maybe some CBGA that wasn't fully decarboxylated.
Thank you for the presentation. Just a quick question. Why the particular focus on IL-8 keratinocyte? Why not IL-3?
Yeah, so there's a lot of different inflammation markers within the skin and the skin assays. But if you hit IL-8, then that's kind of like the key indicator that you are going to see an anti-inflammatory response. There's others like IL-6, et cetera, but the assays for those are not as robust. And so that's why we picked IL-8, because it's a much more robust assay. And if you hit IL-8, then you're pretty assured that you're going to see an anti-inflammatory response in skin versus some of these others where you might get a false positive or a false negative.
Go ahead, Kevin.
I'm sorry.
Excellent talk. You're a good speaker. So I'm just trying to clarify in my own mind. So you're producing CBGA, but the yeast are producing CBGA, and then you're decarboxylating.
Yes, good clarification. The yeast also, just like the plant, produces the acid form. But because of the way that we produce, we are able to easily purify to keep the acid form if that's what we want, or we can decarboxylate it into the neutral form.
So if that's the case, why no research on CBGA, or have you done research?
We have done research on CBGA as well.
Okay.
Yeah, it's just we launched CBG first. So
Can
that's
you
what
enlighten us on any of the CBGA research?
Yeah, so CBGA was, for skin, it was more active for hyperpigmentation and ROS support. Looking at the receptors, it focused more on the pain receptors.
Thank you.
Yeah.
I had a feeling he might ask my question, so I wanted him to go first. I also have a question that was similar to that, but I also wanted to know have you guys done any comparisons with CBG isolate or have you been able to do those type of comparisons, and what did you find? I know it depends on
So you mean like from plant?
From a plant, yes,
Yeah.
the comparisons.
Yeah, so we did, and they're similar. They're basically the same.
Thank you.
Yeah. Well, but I'll use the caveat, as long as it's a high purity plant isolate, yeah. Well, great. Well, thank you so much. I look forward to